Hey there, welcome to Life Noggin. It's me. How well do you remember the 90s? Aside from the questionable fashion choices, this decade gave us some iconic movies like Jurassic Park, which changed pop culture forever, and made us all wonder if it was possible to bring dinosaurs back to life. But how accurate was that movie really? And more importantly, can we really clone a dinosaur? To answer this question, we need to look at how DNA works, how it degrades over time, and some pretty modern breakthroughs in genetics. So let's dive right into Jurassic Park's Dino Dreams. The very first Jurassic Park movie came out in 1993. But before that, people were fascinated by dinosaurs. Mads Mikkelsen brought the iconic villain Hannibal Lecter to life in 1991, and author Michael Crichton wrote his novel Jurassic Park in 1990. But the movie is what popularized the idea of bringing dinosaurs back through genetic engineering, and it did so based on real science. In the movie, InGen founder John Hammond creates a theme park full of cloned dinosaurs by extracting dinosaur DNA from fossilized mosquitoes trapped in amber and piecing together their genome with the help of frog DNA. When it comes to the idea of dinosaurs being brought back to life, this is about as good as it gets for accuracy. But when it comes to the reality of the science behind the movie, we run into our first problem. DNA is a pretty delicate molecule. And once an organism dies, that DNA begins to degrade. Depending on the environment, the half-life of that DNA can vary. But according to a study published in 2022, the half-life of endogenous DNA ranges from 1.3 years to 6.5 million years. This means that no matter where you find it, ancient DNA won't last forever. One of the oldest pieces of functional DNA ever found was extracted from a fossilized beetle that had been trapped in amber for 120 million years. Scientists were able to piece together about 2% of its genome and compared it to closely related living species to determine that this beetle belonged to a new family of true bugs called Siphonaptera. Not only does this show that DNA can last a crazy long time, but it also shows that using DNA from fossils to identify new species is possible. However, even this ancient DNA is degraded. Over millions of years, scientists have developed techniques to sequence older and older fragments of DNA. For example, one research group was able to sequence ancient DNA from a woolly mammoth that lived between 40,000 and 60,000 years ago. According to a study published in 2023, they used shotgun metagenomics to sequence 3.3 gigabases of nuclear DNA from both the permafrost and sediment samples. What this means is that they weren't trying to sequence the entire genome, but rather just parts of it. But for most dinosaurs, even these advanced methods wouldn't work. The reason why the team was able to sequence parts of the mammoth's genome is because they had access to preserved DNA. According to the researchers, the woolly mammoth sample contained at least 1.5 to 2.5 nanograms per microliter of total DNA, which is equivalent to 3 to 5 nanograms of ancient DNA per sample. Meanwhile, the degradation rate of mammoth DNA was estimated to be 0.9 to 1.8% per year. At this rate, after 1 million years, less than 1% of the original DNA would remain. So the fact that any of this mammoth DNA remained after all this time is kind of miraculous. Now, you might be wondering, what about the dinosaurs? Well, thanks to the fossil record, we know that non-avian dinosaurs went extinct around 66 million years ago. This means that any remaining dino DNA would be at least 66 million years old, possibly much older. And according to the woolly mammoth degradation rate, any DNA older than 66 million years would have degraded beyond recognition. There is hope, though. While DNA is incredibly fragile, other molecules like proteins can persist for millions of years. Take, for example, a study published in 2023, which found the oldest proteins ever discovered. These proteins were found in dinosaur eggs from the Upper Jurassic period, which is between 164 and 145 million years ago. The proteins were found on eggshells from 3 different dinosaur species, and they were so well preserved that researchers were able to characterize their amino acid sequences. This discovery suggests that proteins could survive in fossilized eggs for tens of millions of years. Another study published in 2023 took things a step further. This time, researchers were able to extract blood cells from dinosaur fossils that were 76 million years old. The blood cells were found in fossilized dinosaur poop from the genus Boreopelta Mark Michelli. and according to the authors, this is the first report of blood cell preservation in the lacustrine period. So, while it's highly unlikely that we'll ever be able to find intact dino DNA, we may be able to find bits and pieces of their genomes in preserved proteins or blood cells. But finding the genetic material is only half the battle. We'd also have to figure out how to put it together in a way that makes sense. Take the mammoth example again. Even though the team was only able to sequence bits and pieces of its genome, it was still enough to reveal that this particular mammoth was a hybrid of an Asian elephant and a woolly rhinoceros. This suggests that mammoths interbred with other large mammals, which surprised the researchers, since previous studies indicated that mammoths diverged from other elephant relatives earlier than previously thought. This is just one example of how sequencing small bits of ancient DNA can still provide us with useful information. However, putting together a dinosaur genome would be much more difficult since we don't have anything to compare it to. But there may be a loophole. You see, birds are considered to be the direct descendants of theropod dinosaurs. In other words, birds are dinosaurs. And according to a 2023 study, birds also have dinoproteins that don't degrade. This means that instead of trying to piece together the genome of a Tyrannosaurus rex, we could try to piece together the genome of a chicken or a peacock and get a similar result. And this brings us to some recent breakthroughs in genetics. As you probably know, scientists recently used CRISPR technology to edit the DNA of human embryos. But they aren't the first ones to use this tool. One research team was able to use CRISPR to edit the genes of an extinct animal, specifically a passenger pigeon. This experiment took place in 2022, and the goal was to revive the species by bringing it back in domestic chickens. This extinct bird went extinct in 1914 due to overhunting and habitat destruction. And according to the authors of this study, passenger pigeons were the most abundant bird in North America, with flocks numbering in the billions. But today, there are no living specimens left. In order to bring it back, the team needed to edit the DNA of domestic chickens. And according to the study, they created the first ever gene-edited birds with multiple selectable adult phenotypes. They edited 11 genes that were different between the passenger pigeon and the domestic chicken. For example, they changed genes that affected the bird's color, shape, and size. And according to a separate study published in 2023, scientists were able to use a similar method to bring the woolly mammoth back to life. Okay, technically they didn't bring it back in its entirety, but they did bring some of its traits back to life in elephants. As part of this project, the team inserted 2 genes that controlled hair growth and thickness into elephant cells. The scientists then used CRISPR to edit the elephant cells so that they turned off their own genes that controlled hair growth and thickness and replaced them with the mammoth genes. According to the researchers, this resulted in hairy elephants. In other words, scientists have figured out how to use gene editing to bring certain traits back to life in other animals. And while this doesn't mean that we've figured out how to bring extinct animals back to life, it does mean that we're getting closer and closer. But should we bring them back? That's something that experts debate all the time. Bringing back extinct species is known as de-extinction, and it's become a real ethical dilemma for scientists. Take the passenger pigeon example again. Even though the birds brought back to life in the previous study had the physical characteristics of the passenger pigeon, they would have had no idea how to behave in the wild. And since there are no longer any living passenger pigeons, there was nobody that they could learn from. This is a problem for any species that goes extinct. If there's nothing left to learn from, then they'll never truly be reintroduced into the ecosystem. In other words, even if we could bring back a dinosaur, should we? Perhaps some of you watching this video want to bring back the dinosaurs and others don't. And that's okay. The future of de-extinction will likely come down to this question. Maybe one day we'll walk beside a real Tyrannosaurus rex, but until then, we can appreciate the DNA that still lives on in every bird. So do you think we should bring dinosaurs back to life? Let me know in the comments section below. So I'm glad that you watched this whole video. It means a lot to me. And if you want to keep watching, click this video on the left. And if you want to help us make more videos like this, you can join my team by clicking the link in the description. Until next time, remember to stay curious.